Assalamu alaikum dear students. In this pandemic situation, I hope you are well by the grace of Allah. Welcome to our today's online classes. First of all, I would like to introduce myself. I am Sifat Firdausi Shaun, lecturer, Department of Biology, Milestone College. Today, I will discuss about asexual reproduction and parthenogenesis from chapter 10 in your botany book. Let's start our today's class. What we will be learn from our today's lesson. First of all, what is asexual reproduction? Types of asexual reproduction like spore formation process, vegetative reproduction, parthenogenesis, difference between sexual and asexual reproduction. First of all, I would like to definition of asexual reproduction. The reproduction of a plant without the uniting of a pollen and ovule is called asexual reproduction. Asexual reproduction is often referred to as vegetative propagation since no seed is involved in the formation of the new plant. It is known as a clone. Leaves, stem or roots may be used to grow a new plant. Reproduction is actually two types, asexual reproduction and sexual reproduction. Today I will discuss about asexual reproduction like fission, budding, spore formation, regeneration, fragmentation, vegetative propagation. It's all our asexual reproduction. First of all, I will tell you about spore formation. Here you can see this is the picture of terrace and some spore formed in the leaves. The method of spore formation occurs in both unicellular and multicellular organisms. This process takes place in plants. The spore formation, the parent plant produces hundreds of reproductive units called spores in its spore case. When this spore case of the plant bursts, these spores travel in air and land on food or soil. Here they germinate and produce new plants. Example, fungi like mucor, terre's, etc. are example of spore formation process. Then we go for vegetative reproduction. The production of new plants from horizontal stem, plantlets or underground is type of vegetative reproduction. Here you can see this is the figure of a stolon. Stolons are long, trailing, horizontal stems that produces roots. Vegetative reproduction again two types. One is natural vegetative propagation and another is artificial vegetative propagation. Natural vegetative propagation is like it can be propagated by roots or stems, leaves, bulbs. Stem also have three types: underground stem, subaerial stem, aerial stem. And artificial vegetative propagation are cutting, layering, grafting, micropropagation. These also are artificial vegetative propagation and I will discuss about it in these classes. First of all, types of vegetative propagation. Natural, the plant will make copies of itself using structure like bulbs. There you can see this is the picture of daffodil bulbs. And this is the vegetative propagation. It might be artificial. Gardeners can make new plants in a new way the plant is unable to do naturally. For example, taking cuttings. Natural vegetative propagation by stem. Some plants reproduce by means of stem, straggling and branched rhizome reproduce by vegetative reproduction. Example, banana, ginger, turmeric. They reproduce by stem. Here you can see some picture of banana, ginger and potato. Stem tubers are found in potato. Tuber of potato is a swollen apical part of an underground stem. Branched bears a number of nodes or eyes. Each eye bears one or more buds. New plants are produced from the buds on the eyes. The potato crop is raised by tubers, not by seeds. You have to remember this. Some plants reproduced by stem like onions, daffodils and strawberries have stems that can start new offsprings from their stems. Here you can see the picture like runner, rhizome, bulb, corm, tuber, sucker. So some of them are produced by stem. And now underground modification stem like turmeric and ginger reproduced by rhizome, potato is reproduced by stem tuber, onion is reproduced by bulb, gladulus, saffron is reproduced by corm. And then subaerial modification of stem. Here you can see this is the runner, this is the stolon, and this is the adventitious roots, and this is the offset. So grass, centella, strawberry are reproduced by runner, mint, that means pudina, is reproduced by stolon, water hyacinth is reproduced by offset, banana and pineapple reproduced by sucker. And then aerial modification of stem. Watermelon, pumpkins are reproduced by stem tendrils. Here you can see this is the stem tendrils, this is the stem tendril, and this is the bulbil. Here you can see this is the southern part of bulbil. So this is the picture of aerial modification of stem. Now I go for by roots. Some are reproduced by roots like sweet potato, begonia, dahlia have swollen roots that serve as a storage structures. Tubers can develop into new plants that have identical genetic makeup as the parents. Tubers with buds at the base of the stems can also be separated by new plants produced from them. Here you can see this is the picture of old stem and new shoot from bud on the root tuber and tuber. This is the swollen root act as a storage structures. And then some reproduce by leaves. This is the very known for you. Some leaves like begonia have buds on their margins. Here you can see this is the margins. These buds have adventitious roots. Usually when such leaves touch the ground, new plants develop that grow into independent plants. They can be cut and planted into new plants. Here this is the bulbous. I think this is the new for you. In some plant species, the vegetative and floral buds are modified into condensed branches. They store food materials. These modified buds are called bulbous. They help in vegetative propagation. Example like Dioscorea bulbil present in the axil of the leaves. Here you can see this is the axil of the leaves. And then fragmentation. In fragmentation, parental body breaks into two or more fragments either by wave action or by death and decay of older parts. Multicellular organisms with simple body organization such as filamentous algae, Spirogyra breaks up into two or more small pieces or fragments upon maturation. Artificial vegetative reproduction. This is grafting. Here you can see a picture of grafting. This process involves joining the shoot system of a plant that is known as scion to the root system of another plant known as stalk. This is the scion and this is the stalk. The two are joined where the sizes or diameter of the scion and stalk are very close. They are both cut diagonally and placed facing each other. They are then tapped and left to heal with time. Then this is the another artificial vegetative reproduction, that is budding. Budding is the transfer of a bud from one plant to another plant. You can do budding between different cultivars of the same species and in some cases between different species. Unlike grafting, which attaches the entire upper part of a plant, budding only attaches the bud to a different plant. Here you can see this is the tea budding, which is most common method for the propagating fruit race. Now I will go for another artificial vegetative reproduction, which is cutting. Plants such as roses and sugarcane can be cut at the points of nodes and internodes. These points are then placed in moist soils. After some time, adventitious roots develop at the points and new plants begin to grow. There are many types of cutting technique grouped into three categories, that is stem, leaf and root cutting. Stem cutting include hardwood, semi-hardwood and softwood cutting. Leaf cutting include leaf blade cutting, leaf vein cutting, leaf bud cutting and leaf edge cutting. The third category is root cutting. Here you can see this is the picture of basic stem cutting, and this is the basic root cutting and basic planted root products from new shoot. Now is layering. In some plants like raspberries, the stem can be bent and buried in the soils around the plant. After some time, adventitious roots develop of the stem. This part of the stem is known as the layer. The stem can then develop into a new plant. Here you can see so many types of layering. There is mount layering, air layering, types of layering is so many. Simple layer, mound or stole layer, trench layer, tip layer, air layer, so many types of layering is present. And then tissue culture, this is the very modern technique. In this modern technique, tissue scraped from the parent plant is placed in the special growing medium and under certain conditions. The tissue is nourished from the nutrients in the medium until new plants are produced. Orchids, chrysanthemum and asparagus are examples of plants cultivated with tissue culture. And then now this is our another topics. This is Parthenogenesis. Parthenogenesis is come from Greek word. From the Greek Parthenos, that means virgin, genesis means creation. Parthenogenesis is a reproductive strategy that involves development of a female gamete without fertilization. It occurs commonly among lower plants and invertebrate animals, particularly rotifers, aphids, ants, wasps, bees, etc., and rarely among higher vertebrates. These parthenogenesis are two types. One is natural parthenogenesis and another is artificial parthenogenesis. So, natural parthenogenesis. In certain animals, the parthenogenesis occurs regularly, constantly and naturally in their life cycles and is known as the natural parthenogenesis. Natural parthenogenesis again two types, that is haploid or arhenococcus parthenogenesis and diploid or thylacoccus parthenogenesis. Artificial parthenogenesis. The eggs, which always develops into the young individuals by the fertilization, sometimes may develop parthenogenetically under certain artificial conditions. This type of parthenogenesis is known as artificial parthenogenesis. The artificial parthenogenesis may be induced by various chemical and physical means. And now, what is the importance of parthenogenesis? Number 1, parthenogenesis helps in determining the sex of an individual in honey, bee, wasp, etc. It supports the chromosomal theory of inheritance. Number 3, variations from populations are eliminated by parthenogenesis. Number 4, it is the simplest, most stable and easy process for reproduction. Number 5, polyploidy. So, we should know about what is polyploidy. Polyploidy is a condition in which the cells of an organism have more than 2 paired sets of chromosomes. Polyploidy in organisms is caused by parthenogenesis. Number 6, it helps in the development of advantageous mutant characters. Number 7, non-adaptive combination of gene is controlled. Number 8, there are no sterile races. So, this is the significance of parthenogenesis. So dear students, this is your homework. Difference between sexual and asexual reproduction. Number 2, significance of sexual and asexual reproduction. So dear students, I hope you all understand our today's topics. If you have any query, please ask your subject teacher. Thank you for being with me. Stay home and stay safe. Take care. Allah Hafiz.